Good afternoon, everybody. At least afternoon if you happen to be in my time zone in the UK. My name's Andy Johnson. I'm from the University of Derby, and I'm one of the conveners of this meeting. I'm joined in that role by Jean-François Coudenec, who's also at the University of Derby, and by Liz Harper from the University of Cambridge, and Richard Twitchett from the Natural History Museum in London. Liz and Richard will be chairing the sessions before and after the short break midway through. The purpose of this meeting is to discuss the question of whether the ecological niches of species are stable or labile, in other words, whether they change through time. Which is a fairly vacuous question expressed in those very simple terms, because there are examples of both stability and lability. The question is, what's their relative frequency of occurrence, and are there any particular associations, let's say, of lability with particular taxa, particular habitats perhaps, or is it perhaps something to do with the timescale over which studies have taken place. And you're going to come across later on this afternoon examples of very long-term niche stability in some marine plankton over several millions of years. So a particular example recently published is the work of Antel et al. By the way, the references that I'm going to mention in this introduction, I'll be putting into the chat, so there's no need to jot them down now. So that evidence of long-term stability is matched by some evidence of Salpe et al. Of long-term stability in the climatic niche, the thermal niche of marine bivalves and gastropods in the Western Atlantic, again over vast time periods, several million years, and over multiple glacial interglacial cycles. On the other hand, my own work is on bivalves on the eastern side of the North Atlantic, where I see evidence of substantial lability, in the sense that taxa that occur in quite different thermal settings at present occur together in the Pliocene about 3 million years ago of the eastern North Atlantic area. So on the face of it, therefore, quite substantial lability in one group of species or another. That's something we're investigating, which of it is, by independent means of establishing temperature using oxygen isotope thermometry. So indications of lability as well as of stability over very long timescales. And now if I just switch to if I share my screen, oh, it's disappeared. No, it hasn't. There it is. So you should now have the poster for the, whoa, it's gone too far. You should now be seeing the poster for the event. And if perhaps you're wondering what the significance is of a rather attractive bird in the top right corner, it's a very well-known example of an invasive species, which are a source of evidence of quite substantial niche change over short time scales. The particular species you see represented there is a ring-necked parakeet, seen in its native habitat in the Indo-Gangetic plain. That particular example is photographed in Pakistan. That species now occurs all over, well, not all over, but very widely throughout the world. It's very common in Europe. It's exceedingly common in parts of the UK, and extends right up as far as Scotland. So, anybody who's familiar with the climate in Scotland will know that it's not like the climate of the Indo-Gangetic plain. So, a good example, therefore, of niche change. Now, there are estimates of as much as 50% of invasive species showing niche change, particularly in terms of their climatic niche or their thermal niche. But that's been recently disputed by Liu et al. In a paper in 2020. But nevertheless, there are examples amongst invasive species of substantial niche change. So, but that's just invasive species. That's not all species. We need a more general picture than that. And we need some idea not only of the frequency, but of the occurrence. Are there, as I've indicated before, associations with particular taxa, habitats, and so on and so forth. And I think I hardly need to say that this is of importance both to, let's say, paleoecologists, people who want to try and infer the environment in the past on the basis of fossil assemblages by reference to the habitats and habits of modern examples of the species represented. And it's important, of course, for conservation and management purposes. If niches can change, if species can change their niches, obviously the way that we might manage them in the future in the context of environmental change would be affected. So before I pass over to Liz Harper, who's going to chair the first session, can I just mention a few housekeeping points, which you should now see on the slide that's on the screen now, concerning the fact that your microphone will be muted and camera will be switched off. So you will only be able to communicate by the chat and the question and answer box function. There is a facility, as you'll see there, for closed captions if you press the CC button. As it says at the bottom, the video is being live streamed and recorded on YouTube, and it'll be available in a few days on the Geological Society's channel. So if I could now hand over to Liz Harper, who's going to chair the first half of the session. Liz? Right,
thank you. So I would like to add my welcome to everybody for coming to this seminar, and just sort of slightly out of text as well, to thank Andy Johnson very much for getting this conference together. But I think without further ado, I'd like to introduce Annarizko, who comes from the National Museum of Natural Sciences in Madrid. And she's going to be talking to us about ecophysiological responses to changing environmental conditions in marine invertebrates, molecular keys to adaptation. So thank you very much, Anna.
Thank you. Let me share my screen. Thank you very much for the introduction. I would like to start by thanking Andy for the invitation. This gave me an opportunity to be in contact with my British colleagues again. I was at the Natural History Museum in London for 5 years, and I still retain a bit of an affiliation there. And I very much miss the UK and my colleagues there. So this was a great opportunity to catch up a bit. And I feel like a bit of an outlier here because I'll be talking about molecules. So I'll try to not get into much detail of genes and stuff like that to not get you in like a sleepy state. But I hope it's interesting and nice for you to see this. So obviously in this meeting, I don't have to tell anybody the effects and the consequences of climate change. I know everybody much better than me knows this, but just a brief reminder that we know way more about the effects of climate change and the consequences in terrestrial habitats than that we know of what is going to happen or what is happening in the oceans. And pretty much everything, not everything, but almost all the reports and the articles that we see are related to coral reefs, because obviously these are key and iconic ecosystems, but these are not the only ecosystems or the only organisms in the ocean. But we know that around 40% of the oceans have been affected already by climate change and by anthropogenic disturbances. Sorry. And what does it mean? Well, it means that there are currently shifts in species community structure. So the communities of different species are changing, and I'll show you just an example. There are shifts in species biogeography, and I think people here know this much better than I. But there are other issues related to climate change in the ocean, like ocean acidification, which is massively, is a dramatic situation for some organisms, like those that build their skeletons with calcium carbonate. And of course, there's coral bleaching, which you have an example here in this picture, and affects corals and the symbiotic association they have with zooxanthellae and Chile. But all this comes down to biodiversity loss, and this is a problem because we know very little of the biodiversity that we have in the oceans, and especially in ocean or in places in the ocean that are very remote and difficult to access, like the deep sea. So in my field, we study the ecophysiology or the adaptations, molecular adaptations in general, to the different climate or different conditions, environmental conditions. And in the past, or right now as well, but there are many different ways of studying this. And one way is like doing correlations between phenotypic traits and environmental characters. And of course, there is experimental manipulation, but this is science fiction for the organisms that I'm going to present to you, because this can be done mostly with vertebrates or with model organisms, but not with the organisms that I will introduce you to. But there is something that is transformative right now, and this is the field of genomics, which allows us to understand the transcription, the genomic responses of animals, the genomic adaptations, or the traces that the evolution left in our genomes, and how we adapted to different conditions across time. And I'm going to specifically talk about transcriptomics, which is the part of the genomic field that deals with the genes that are expressed. So anything that is expressed is captured by this sequence technology, and this allows us to understand the changes in expression when something is different in the environment. We can identify molecular pathways that are important to adapt or to respond to these climatic, environmental conditions. We can also see genes under selection, and we can correlate this with environments. And as I was telling you before, most of what we know comes from the field of coral reef ecology and physiology. But I'm going to show you that even in coral reefs, not everything are corals. And I know there are some people of the mollusk field here, and of course, most of what we have in a coral reef is a mollusk. But we also have other things like sponges and many other organisms that are usually poorly known, because corals we know they're beautiful, they're massive, and they're extremely important for these ecosystems. But right now there are processes happening in some tropical coral reefs that are affecting the corals more than, for instance, the sponges. And you have a picture here of a coral reef in Bocas del Toro in Panama, and pretty much everything you can see is a sponge. So now we have to call them sponge reefs. And I'll be talking a little bit about why this is happening today. So I'm going to present you this beautiful, to say something, beautiful species that we've been working on. So the first two, the ones that are on the top, these are two sponges. One is Isodictia, which comes from the Antarctic, and the other one is Spongia officinalis, which is the common bath sponge, responsible for us bathing with sponges across time, and it comes from the Mediterranean. And in the bottom, we have Nemertean, a ribbon worm, also from Antarctica, that's called Antarctonemertes valida, and a sea urchin called Arbacia lixula, which is very common now in the Mediterranean, but it came from tropical, we think tropical Brazilian coasts. So it's a recent colonizer in the Mediterranean. We don't call them invasive, although it kind of like is a bit now. So it's in the process of adapting itself to the Mediterranean environment. So our question was how marine invertebrates cope with thermal shifts at the transcriptional level, as I was telling you, by looking at the transcriptomes of these organisms. And we were interested in these two settings, the Mediterranean and the Antarctica, because they are two of the most threatened marine areas by global warming. So the predicted temperature by 2050 in the Mediterranean will be an increase in 1.5 degrees, and in Antarctica this will happen, or this is predicted to happen by 2044. And this is a huge change for most of the species, and we don't know what it means because there are very, very few studies on this subject. So what we did is a similar or relatively similar setting in all species, but I will walk through I will walk you through the different experimental settings. So the first study that we did with two Mediterranean species, the sponge and the echinodon, the sea urchin. In the case of the sponge, we always had to put them in aquaria for about 2 days or 1 or 2 days to acclimate them, because they usually don't feel very well if you start experimenting with them right when you put them in the aquaria. So you have to give them a little time to relax. And then we started the thermal injury. So in this case, we exposed them to 3 different temperatures progressively. The good thing about this experiment is that it was like a thermal wave, one of these heat waves that happens in the Mediterranean. So we first exposed them to 27 degrees and then to 30 degrees for 1 hour, which was a short-term exposure. And then we kept them at 30 degrees for 2 days. In the case of the sea urchin, we didn't expose it only to warm waters. We also exposed it to cold waters because, as I told you before, it has tropical it has a tropical origin. So we were interested in knowing how this sea urchin adapted to the Mediterranean, where it can get really cold in the winter, especially in areas where this is a very common sea urchin, like in the coast of the Western Mediterranean. So we exposed it to 22 degrees and then to 7 degrees in about 2 days. And in the Antarctic species, you may know that it's a little bit trickier to work with them because you have a very short time frame and everything has to work in very extreme conditions. So it was a little bit harder. In the case of the sponge, we were able to put them in controlled settings for about 2 days and then expose them to 3 degrees and then 5 degrees. Usually in the South Shetlands, where we did the experiment, these sponges, the water can get up to 3 degrees sometimes. So this was not a huge or an extreme condition for them, but it can never get to 5 degrees. So this was our extreme temperature. And we also took advantage of, this was not experimental, it was a natural setting for the ribbonworm. We took advantage of the situation of our base, the Spanish base in Deception Island, which is an active volcano, and it has fumaroles. It has active fumaroles, and we are not the only ones taking advantage of this natural setting. As you can see here, is a very popular site for tourism, where everybody goes and enjoys bathing in Antarctica. So these settings they're not only very hot, they can go up to 10 degrees, but they also have sulfites. So we sampled the ribbonworms there, and actually these were the only megafauna that we could find, together with a gastropod that we couldn't identify. So they were the only ones living in there. So we compared the transcriptome of these guys living in these hot conditions with the transcriptomes of other ribbonworms from the same species living in normal conditions in Antarctica. So the first thing that you have here, this is what we call a heat map, and every single line that you have here is a gene and the expression level that will go in this case in the left will go from very low expressed in violet to very highly expressed in yellow. Okay, and these are the different treatments. And in this other heat map, it's a similar situation, but very low expressed is blue and very highly expressed is red. And I just wanted you to see that this means that lots of different genes are responding to these treatments, okay? So the thermal injuries in these organisms, and these are two sponges, they cause a huge transcriptional response. So they're getting a little crazy with these treatments. And this is just a summary of what happens. And we did this for the Mediterranean sponge. And it's very similar to what happens in the Antarctic sponge, but with a little twist that I will tell you later. So basically what happens in the beginning when we expose them to 27 degrees is that there is a cellular response to DNA damage, because they have to protect their DNA in these situations, because otherwise they are they're not going to make it, and there's a lot of apoptotic signaling pathways happening. So there's cell death. The sponge is shedding lots of death, and it usually has like even necrotic tissue, so it's not doing very, very well. If you expose it to 30 degrees coming from these 27 degrees, the response is even tougher for the sponge, and it usually kicks in the classical response to heat, which involves the heat shock proteins, which are proteins that are responsible for the correct folding of other proteins, because in hot conditions, proteins are usually not folding right, so they degenerate, and they have to take either just remove them or try to fold them correctly. And there's also an immune response happening, because most of many of the symbiotic bacteria that sponges have, and they're in the water, can turn pathogenic in these very hot conditions. So they have to make sure that they're not affected by these pathogens. But if you leave them for 2 days in this temperature at 30 degrees, they turn to the normal activity, physiological normal activity, and regulation of developmental processes to regenerate all this necrotic tissue or this tissue that died before happens, and they turn down the inflammatory response, they turn down the immune response, and they go back to normal. And this was a bit surprising, but when we thought about it, and when we were doing a little bit more of a literature review, it's not that surprising if we think that sponges have been here with us, not with us. They have been here way before we came in, for 600 million years at least. So they have seen these changes in the temperature of the seawater many times. So they obviously have these mechanisms to cope with the changes. But this is not something that happens in sorry, I just wanted to show you here that this is the classical pathways for these heat shock proteins, and these are upregulated, and it happens both in the Mediterranean sponge and the Antarctic sponge. But one thing that was interesting is that in the case of the Antarctic sponge, instead of using one of these heat shock protein 70s, it expanded the complement. So it is more able to cope with this stress that happens when there's a thermal increase than the Mediterranean sponge. But the interesting thing is that if you keep them at higher temperatures for longer, they're not able to cope like the Mediterranean sponges, and they die. And this was surprising and concerning as well, because this means that tropical and temperate waters with sponges will turn, like the coral reefs, will turn into sponge reefs because they're doing great. They're doing much better than the corals. They have mechanisms to adapt. However, these settings that we see here in cold waters like Antarctica, or even in the Arctic, where sponges are the dominant, massive, and super abundant organisms structuring the communities, these can totally shift, and they can the communities can change massively, and this has important cascading effects on the entire ecosystem, obviously. But what happens to the other two organisms? So the responses were similar, but the interesting thing about the sea urchin was that it didn't respond to the thermal increase. They were doing absolutely great in hot conditions. It was responding to the cold conditions because, as I told you, it's a originally tropical sea urchin. So the cold was not fantastic for them, and of course the ribbon worm was responding to the increased temperatures. But I'll show you why. In the case of the sea urchin, it was a very similar response with heat shock proteins, cell death, and immune responses all upregulated, but in the case of the cold. And this has massive repercussions in the ecosystems, because as temperatures increase in the Mediterranean, these populations of sea urchins are out of control and they are blooming. These urchins have massive populations and they have barren effects on the ecosystems, like pretty much clearing up everything in the rocks in the Mediterranean. And in the case of the ribbonworm, I told you that it is responding to the conditions of temperature, but the truth is that it doesn't respond to the temperature, to the hot temperature. It responds to the sulfides by upregulating the detoxification pathways and lipid metabolism and mitochondrial respiration, which is what happens with hydrothermal vent organisms. And only the neurogenesis pathways, so the way that they make their neural system, is responding to temperature, because this is something that everything in every organism happens. So that was the only thing that was a little bit tweaked in the ribbonworm. But other than that, it was doing absolutely great. And this was actually, I mean, it was what we saw in the reality, because they were doing fine in these ecosystems. So we are in a situation of winners and losers. So this will, of course, this is just a very brief snapshot of a few organisms, and this cannot be extrapolated to everything that happens in other organisms in the ocean. But this speaks to us as we have to continue understanding what happens with these organisms, because we don't know anything, and many of them will be doing great and will be expanding their populations and will be changing their abundances, but many of them will be lost. And this was all that I have to tell you, and thank you very much again for the invitation.
Well, thank you very much, Anna. That was a really interesting talk. So I'm hoping that to invite people to ask questions in the Q&A box. So just while people are getting brave and perhaps typing, oh, somebody's come already, so we better have a look at that. Okay, so Dave Horn has said, thank you very much. And how do you determine the optimum conditions and the duration of acclimation for the organisms, which is what I was about to ask as well.
So basically, before doing these experiments, we do a round of preliminary experiments to see if they die and how long we can keep them. And usually we go and measure some physiological factors, like, I don't know, in the case of sponges, if they keep pumping and how often they pump, and if they have the oscula open and things like that. And in the case of the ribbonworms, I mean, they were in the natural setting, so that doesn't matter. And in the case of the sea urchins, if they were responsive and on all the stuff. And once we know that we can have them in the acclimation going back to normal activity for 1-2 days, then we start the experiments.
Just while we wait for somebody else to ask a question, I was quite interested in this sea urchin that's come from Brazil. Do you know when it came? And I wonder what kind of temperature changes it might have experienced in the Mediterranean since it's been there. Has it been there a long while?
I actually don't remember, and this is something that I was expecting, and I thought I had to look at when it came. But I honestly don't remember. But the truth is that it's expanded across the Mediterranean. So it is exposed to a wide variety of temperatures, from 7 degrees in the winter in the Western Mediterranean all the way to 30 degrees in Greece in summer, and it's everywhere. So I guess it doesn't have much of a problem adapting to these conditions.
Okay. Just waiting for people to be brave and ask questions. Can I ask about the Antarctic ones again? Oh, Andy Johnson's going to ask a question. Perhaps we'll let him ask a question. Welcome, Andy. No, I'm not sure that Andy is asking a question after all.
Yes, he is, because he's now unmuted himself.
Oh, good.
Sorry. Anna, I'm just wondering, is there the examples of long-term spaces that I referred to in the introduction are in protists, basically. You know, they're single-celled animals and plants. Is there any reason for thinking that there might be a difference in response from the somewhat higher organisms, albeit the sponges aren't that much higher, compared to them in their ability to, or in the ways that they would respond to heat shock?
Well, the truth is, I don't think they have all this complement of heat shock proteins, the protists. So they will for sure have other ways of coping with changes. I don't really know, but the truth is that from the beginning of metazoans, and you know, sponges are at the beginning of the of it, they do have very similar complements of heat shock proteins and immune system pathways. So they're very I don't know. They have similar ways of coping, but I don't know about the protists. I don't think they can cope as well.
Okay, interesting.
Okay, well, I think we've probably got time for one more question before we have the next talk. So Ben Moon has said, can the rate of change in conditions lead to a difference in the reaction, or is it principally or primarily the magnitude that's important?
No, you're absolutely right. It's both things. So if you expose them very quickly to the heat shock, they will have a very acute response. But if you do it like over time, I don't know, days or something like that, and you keep them in this condition for 10 days, the response will be a little less. It will be similar, but it will be less acute.
Can I just follow up on that slightly? I know I'm not supposed to be asking another question, but the Antarctic, presumably the rate of change is actually quite slow there, or not?
The rate of changes, what do you mean?
Sort of the experience they might have over several days, the temperature is not going to change that as rapidly as in your experiment.
No, I don't think.
Whereas in the Mediterranean, you were mimicking a Mediterranean heat wave, yeah? Mm-hmm. Yeah, okay. Thank you. Well, I think we probably better move on now, if that's okay. So now we are calling upon, thank you very much, Anna, I should say, thank you, that was really interesting. So now calling on Rachid Cheddardi from the University of Montpellier in France, who is going to talk about niche shift or not for some major European tree species over the Holocene. So thank you very much.
Can you see my
yes,
screen?
We can. Yes, we can.
Okay, good evening, everybody. And thank you, Andy, for inviting me to this session on niche change. I'm paleoecologist. I'm located at the Institute of Evolutionary Sciences, the University of Montpellier in France. And I said yes to Andy to give this talk today because the topic is very interesting, and of course, and also over the past 10 years or 15 years, modelers have produced nice models to predict future changes or future suitable habitats for plant species, mostly threatened plant species. And these models, in fact, there are two types of models: the niche-based models and what we call the dynamic models, which are rather process-based models. The niche-based models have been extremely used over the past decade or 15 years. And I would say there was a problem for me as a paleoecologist because most of the simulations that I have seen were based on the modern range of the species. And I would say also as a reviewer, I was sometimes, how to say, not shocked, but embarrassed by some papers where some colleagues were using these niche-based models in a kind of rough way, just taking the modern distribution of the species, the modern climate, and, if I may, pressing a button and predicting the suitable habitat. So this is, I would say, a major topic for paleoecologists and ecologists sensu lato. So today, I will, my talk will be quite brief, but I will try to show you, give you some slides about the niche change of 3 European tree species, which are spruce, beech, and silver fir. The definition of the fundamental and realized niche have been defined for many years, or decades now. The fundamental niche is the widest range that a species may occupy over time. And the realized niche is a subset of that niche. Recently, Jorge Soberon has, in his paper in Ecology Letters in 2007, identified or synthesized the variables that define a niche. And there are 2 sets, or 2 types of variables: resources, which are biotic variables, like competition between species and other interactions between species, and conditions, which are environmental or abiotic variables, mostly climate, that define the niche of the species. So this is the way we try to tackle the problem of the niche change through time. We had to have a data set that can cover both in space and in time the European space. This is necessary so that we can track the occurrences, proportions of the species in space, but also in time. And the other condition is that for the same time span in which we are interested, we should have not only the species occurrence and distribution, but also the climate under which the species occurs. So this is basically, or roughly, how the data that we have used. Plants or trees release pollen grains in the atmosphere, which are deposited on the ground. They are preserved in the sediments, and these sediments are collected from humid environments like lakes, ponds, swamps, and so on. So once we collect these corings from these humid environments, we subsample them and to date them so that we can have a chronological time frame for the coring, and at the same time we treat them in the lab and analyze their content in terms of pollen grains, but not only pollen grains, but there are other micro-remains and so on. But the idea is to have, for each sample, the pollen content, so that we can track in time the presence of many species. That is one thing, but also the expansion of the species, regression, extension, and some other basic information from these pollen grains. So the data set that is now widely used comes from the European Pollen Database. That it is a public database, and each dot that you see on this map at 9,000 before the present corresponds to a coring or a time series. Just for information, one time series requires something like 1 year work in the lab. So you can imagine the time that was spent by colleagues to make these analyses. So from this data set, from the European Pollen database, we can extract information about the distribution of the plant species at a specific time span, but at the same time, we can have information also about climate. I'm not going to go into the details of how we obtain climate and plant species or the interpolation and so on, but let's just keep in mind that we can obtain these two types of information for any, well, for a certain time slices available in the database. So once we have the species occurrences and also proportions, because it can give us some information about the population density and so on, and climates, and particularly specific climate variable like general temperature in this case for a specific time slice, we can evaluate the climatic niche of the species at that specific time slice. In this case, we have the species, the densest populations occur between -4 and -2, roughly, and sparse populations between 0 and -12. So from this database, we can have information about the species density somehow, and the temperature range, or the span, the climatic span, the temperature range within which the species occurs. So we did this exercise for different time slices between 10,000. I don't know if this is big enough, so you can, but you can trust me. This is 10,000, and this is 3,000, and these are all the time slices between 10 and 3, 8 time slices. This is spruce, Picea abies, and this is beech, Fagus sylvatica, and this is silver fir, Abies alba, the 3 species of interest in this study. So what we can see here, okay, well, the gray boxes are the realized niche for each time slice. The orange one, the orange boxes are the fundamental niche, or the sum of all the niches over the throughout the Holocene. And the green box is the modern range of the species. Of course, this, if you look at this map, for instance, spruce can be found in all this area in red and green, but in red there are sparse populations in terms of climatic amplitude. So what we can see from these climatic niches for the 3 species is that in the first place, the 3 species, each species occupies a different amplitude of climate. What I mean is that, for instance, for silver fir, the amplitude is much lower than for beech. So there are the amplitude of this climatic niche is different between species, and also this niche varies through time. As you can see for spruce here, the amplitude covered by the climatic niche at 10,000 is quite different from the niche for the same species 2,000 years later. And this is almost what we observe for all the European species. The other thing that we observe is that none of the 3 species have occupied the full range that is the sum of what we observe for the Holocene. They occupy only a partial part of this extended range. The third thing, or the last thing that we can observe also, which concerns what I have seen earlier concerning the niche modeling, is that if we look at the modern range of the three species, we can see that they don't encompass the full range of the species. But not only that, but also there is a difference between the modern range and the full niche. In other words, for instance, spruce and silver fir, the range is, in the case of spruce, 4.5 degrees lower than the full range, which means that the species is today located in colder areas than during the Holocene. This is not the case for beech, which is already in the warmer areas. So if one uses only these modern ranges, these modern niches, to make a prediction of suitable habitats, then it might be highly biased. My colleagues, my colleague Luigi Morano did a different exercise using a niche-based model, but at this time he used a different climate simulations for different time slices between 13,000 and today for the same species. And then he compared what he hindcasted with the fossil data. So what we observed is that, in fact, something similar before 7,000, before the present, we had the niche was not only different, but was not in phase with the modern niche. This confirms what we see from the reconstructed climate. In other words, what we observe today in terms of the species distribution for these three species cannot be used to forecast the species range for tomorrow with future climate scenarios. And what he has demonstrated is that, in fact, if the model is calibrated using all these past niches, then the prediction or the projection of the future suitable habitats can be much improved. So to reply to the question I have posed at the beginning, if one uses only the last distribution or the last niche of the species to predict future range or distribution of the species, then it will be biased or even wrong. And so I would like to make just some simple conclusions to this little work we've done on the species niches, is that, in fact, the modern range that we observe today of these three species is also a partial climatic niche. It cannot be considered as the full niche. And also, this is also the case of the other three species. But in fact, this is related to a continuous disequilibrium between the species occurrence in an area and climate. These are long-lived species, and this disequilibrium is more or less marked depending on the velocity of the climate change, competition between species and other and migration rates and so on. So taking the modern distribution and climates of the European tree species as a fundamental niche, which can help us to model future suitable habitats, can be quite biased. Thank you very much.
So thank you very much indeed for that interesting talk. So again, a call for people to put questions in the Q&A box, and I think we've got one already from Jess McCoy. It says, an excellent presentation, very interesting. I was wondering whether you were planning to do similar studies in the pre-Quaternary.
In fact, as I said, all this work is based on the European Pound database, and the deeper in time you go and the less data you have. So going back to prior to 20,000 years before the present, we have very few records, and therefore these estimations will be biased due to the lack of extended data in time.
Right, thank you very much. And there's another one from Paolo Albano. Thank you again for your very clear presentation. And the question is, which aspects of tree life histories may lead to the results you've shown us, which contrast with those for mollusks and forams in the marine realm that would support niche conservation?
What factors differ? Factors other than climate? Oh, well, yes. Well, in fact, there is one thing we have, I would like to stress, is that if there is only one variable that affects the niche, then that is, to me at least, that is sufficient to say that the niche changes. Now, if we, in fact, even for climate variables, I talked about January temperature, but there are many, even in terms of temperature, many other temperature variables that can be important for precipitations, but also they can be other biotic variables, such as I said, competition and between species for light or for nutrients or for any other elements. And now some species-based models take into account competition between species. So I talked only about the abiotic variables, but of course there are many biotic variables that should be taken into account. And to me, those models that are the most pertinent to make these predictions for future suitable habitats are the process-based models, which take into account biotic and abiotic variables.
Thank you. It's a sort of follow-on question for that. It's appeared in the chat from Damien, and it's asking if there could be other factors from climate driving the observed changes, for example, fluctuations in wind strength over time.
Definitely. Trees, mostly mostly trees, which are in either in Europe or in the Mediterranean, grow are in mountain areas, and mountain areas are how to say, we consider them also as refugial areas. In other words, in mountain areas, there is an impact of local elements like wind, insolation, exposure, slope, and so on. And so, of course, there are many other variables, local variables, that can, how to say, constrain the niche of species. That's where speed wind speed is also an important variable also.
Okay. Can I ask a question from me, which is probably showing my ignorance of plants? How fast can they migrate?
No, this is not. This is a very good question, because the problem with the climate change is that the migration rates of, how to say, most of the plants and mostly trees is much lower than the velocity of the climate change. And we did some computation also of migration rates. Let's say they can migrate between 50 meters per decade, between 50 and 250 meters. But of course, migration does not mean that the tree is moving, but this is related to the dispersal of grains and seeds and pollen grains. So the migration rates are, how to say, also species dependent, because conifers, like the ones I have shown, have a much lower dispersal than many other angiosperms.
Yeah. Okay. Thank you. And Ben Moon has asked another question that looks that you could answer it. How does the fossil range and niche represent the temperature range in Europe? And do these species have current or historical ranges outside Europe that might have a broader temperature range?
If the question is about the same species outside Europe?
I think so.
Well, these species are really a typical European species. Of course, there are other, if you take, for instance, Abies alba is one species. There is only one species in Europe. It doesn't occur in Asia or in America. There are other species. And of course, if you look at sister or sibling species in Asia, the climate range can be very different. For beech, if you look at Fagus grandifolia in the US, it has a much wider range than Fagus sylvatica in Europe. But these are two different species, I would say.
Okay. Any more questions? I've had quite a few, so it looks as though you might get away with that then. But thank you very much, and there've been lots of questions there.
Thank you very much.
Which is very good. So thank you very much. So I think we'll move on now. Thank you. And our last speaker in this particular session is Carl Flesser from the University of Arizona, and he'll be talking about niche stability and conservation paleobiology, clams, condors, and cottonwoods. So quite a variety there. So thank you very much, Carl.
Okay, thank you. I'm hoping, Liz, that you can confirm that my slides are up and my
they
audio
are
is
indeed.
Good.
They are
right,
indeed.
Thank you very much, and thank you for that introduction, and thanks, Andy, for putting this program together. Really, 2 points I want to sort of leave you with, and I'll tell you them at the beginning. First of all, from the perspective of conservation biology, niche labelity is a good thing, and I'll give you some examples of where it's proven to be a good thing, certainly on the sorts of timescales that conservation biologists are focused on. And the second point I want to make, and it's sort of evident, I think, in the range of speakers, and I appreciate being involved in this group for the following reason. I want to remind everyone that geological time does include now. All right, so conservation paleobiology, really, the sorts of things that we can do and are doing include a reconstruction of baseline conditions. This is a way of assessing human impact when prior conditions were not evident. So it's a before-after comparison. We can assess reference conditions for habitat restoration, and I'll give you an example of that one as well, especially where space for time substitutions are not possible, where one cannot find unaltered habitats that are restoration goals, if you will. Thirdly, paleobiology of species at risk under pre-impact conditions. Most species at risk, threatened and endangered species, have much reduced population sizes and ranges. And we really would like to know is what were things like when conditions were more advantageous for those species. And certainly looking at the paleobiology of species at risk has a certain advantage in that it is a decidedly non-invasive technique. You're not affecting the living, and that's a good thing. Ideally, one would like to reconstruct the natural range of variability. And Rashid's presentation is a great example of sort of looking into the past to reveal sort of, well, what was really the total range of climatic conditions under which these particular species might have occurred. And this also allows one to look at the effect of rare events and how those might have affected rare and endangered species. I come now to the issue of sort of the challenge of deep time. Most of my work and most of the work of my colleagues in this field have focused on near time events. Going into the deep time, we have problems arising. We'd love to be able to look at deep time and understand better the biotic response to environmental change as a way of predicting what might happen in the future. We'd love to be able to assess species vulnerability by looking at the fossil record. But in fact, there are several problems involved here, two of which are, of course, temporal resolution. The deeper in time you go, the less resolution one has in general. And of course, the problem of no analog ecosystems. From the perspective of someone who works in extant modern environments, no analog ecosystems existed in the past. And for those of you who work on fossil ecosystems, no analog ecosystems exist in the present and in the future. So that's a bit of a challenge. Now, the sorts of things that I'm going to be talking about in conservation biology, conservation paleobiology, have been reviewed many times, and the citation I give there at the bottom of this slide is neither the first word, the last word, or the only word on the subject, but it's a useful place to begin. Conservation biology occurs in a policy context. It is an academic field, but it is an applied field. And in the applied aspect, it occurs in a policy context. And the policies are determined by such things as the United States Endangered Species Act, which I'm most familiar with EU Habitats, Biodiversity and Habitats Directives, in the Australia EPBC Act, and every country, or most every country, has some sort of policy or legislation that determines what happens with endangered species, with threatened species. And this is where the term threatened, vulnerable, and endangered species come from. Most definitions focus on two aspects. First is a decline in population size of some magnitude. And secondly, a reduction in range, or in fact, critical habitat, which is the language of the Endangered Species Act in the US. And most efforts are focused on the issue of habitat, because in practice, conservation biology is a crisis discipline. We want to fix a problem. We want to keep a problem from getting worse. And so it requires application in a very short time frame, and frequently academic studies don't provide the information quickly enough. So it's a crisis discipline in that sense. The focus historically has been on habitat rather than on determining niche dimensions of individual species, but the habitats of those species. And that's a subtle, but I think important distinction. But in effect, practitioners, those people who are trying to restore habitats, to preserve or enhance endangered species, recover endangered species, really can't afford to be very fussy or very patient. I want to give you three stories to illustrate some of the aspects of conservation biology that may be of interest to this particular group. The first story is the one in which I'm most familiar, and it has to do with bivalve mollusks, a particular species of bivalve mollusk, the Mactrid Mulinia modesta. And this particular species was tremendously abundant when the Colorado River reached the sea. It no longer does, as I'll point out in a moment. And the picture in the lower left illustrates one of these condensed assemblages resulting from the accumulation of shells along the shore, resulting from the erosion of pre-existing muds and shells. So they were really abundant. There was a population decline, we've estimated, of about 90% of this particular species as a result of the diversion of Colorado River water before it reached the sea. We think that because the oxygen isotopes in these shells indicate brackish water that is much less saline than at present. Colorado River has not flowed all the way to its mouth since about 1960. And 1960 is when one of the large reservoirs on the system in the United States began to retain a lot of water, and most of the water then became used. 90% of the water is used in the United States for agriculture principally, and a little bit less so for growing cities. 10% of the water, it's a fixed sum, but approximately 10% of the water is delivered to Mexico by a treaty, by virtue of a treaty arrangement. Mexico, what it does with its 10% of the water is exactly what the US does with its 90%. It allocates water for agriculture, and it allocates water for its cities along the border. What you're looking at here is the former channel of the Colorado River, and you're actually looking upstream. This little trickle of water here is actually tidal water, trying to sort of get as far up into the system as possible. But you can get some sense of the extent of the former Colorado River when it still flowed all the way to the Gulf of California. What we've done is to try to get at some of these reference ecosystems and say, well, what would be to bring this clam back to its previous abundance? And what we've done is to look at stable isotopes of oxygen in these shells as a function of distance from the river mouth. And these are the subfossil accumulations of shells, much like the one I showed you. And what this shows is, not surprisingly, is that salinity increased as the distance from the mouth of the river increased. At the present time, present day salinity is about 38 parts per thousand, or 38 PSU, if you will. Excuse me, going the wrong way here. We'll want to try to restore these populations. We would need to get at, we think, to restore the populations that were present at about 20 or 22 kilometers from the river's mouth. We would need about 32 parts per thousand salinity. What would that take? Well, that would take about 10% of the river's annual flow, which is quite a challenge. But nevertheless, there may be sort of ways of getting around that and bringing some of these populations of these clams back. And the reason we might be able to bring these back is that, surprisingly enough, Mulinia modesta persists at much higher salinities than it did in the past. It persists today at about 40 parts per thousand. You need to know where to go to find these things. And it's a bit of work mucking about, quite literally mucking about, in the intertidal flats to find these specimens. But they still exhibit the radical increase in salinity. So this is a population reservoir for recovery, and an illustration of the fact that the niche of this particular species, with respect to salinity at least, was quite labile. My second example is quite different, and it concerns the California condor. This is a really magnificent bird, about a 3-meter wingspan. And as you might tell from this particular image, each and every one of these condors has a number. It's a bit like a race car. You can figure out which one it is by the number that it bears. And they also carry transponders, so you can actually keep track of each individual California condor that's been re-released into the wild. They were extinct in the wild in 1987. The remaining 10 to 22 individuals that were captured were then bred in captivity. And following that, some reintroduction and, technically speaking, some rewilding. So the population now in the wild is about 230. There are about 110 or so, I think, still in the captive breeding program with active re-release. Now, the US Endangered Species Act requires action to recover the population, and this was done through captive breeding, and to protect critical habitat. And one of the fascinating things to me is that you look at the fossil record of the California condor. They are known from such famous localities as the La Brea tar pits, now in the middle of Los Angeles, southwestern caves, which will be really the focus here, but they're also known from fossil deposits in Florida and Western New York, of all places. This is a really quite far-ranging species in the Pleistocene. This species has now been reintroduced to northern Arizona and southern Utah, reintroduced to central and southern California, from which the 10 to or so individuals were captured for breeding. And it's been introduced into Baja California in Mexico. Insights into sort of the past paleoecology of the California condor is really quite interesting. Here's some record of fossil condors from a particular cave in northern Arizona. And the really cool thing is here, this cave preserves also the diet of the dead. Remember, they're carrion feeders. And their diet was the Pleistocene megafauna. In this particular case, these deposits are 10,000 years before present or older. And what you see in these deposits are horses, which became extinct in North America, camels, Harrington's mountain goat, bison, and the mammoth, Columbian mammoth. Now, these particular species did not wander into the caves and die by accident in the caves. Clearly, they were harvested, their carcasses were harvested, and some of their bones and remains were brought back to the cave. And also in these cave deposits, there are some items that might have served as either hors d'oeuvres or desserts for these California condors, or might have simply occurred in the cave quite naturally. So we have some insight into their diet in the past. We also have insight into their diet from carbon isotopes and nitrogen isotopes, from Chamberlain et al.'s work in PNAS in 2005. This is a bit of a complicated slide, so I just want to sort of walk you through the text here and to sort of illustrate what was done. What these folks did was to look at carbon and nitrogen isotopes in feathers and hair from live collected modern and museum specimens of condors, as well as bone collagen in modern, historic, and fossil condors. And basically what they have found is that condor diets have changed through time. The Pleistocene condors had a highly variable diet. Here we have the bones of the Pleistocene condors, including megafauna, as indicated with the cave deposits, as well as marine mammals, beached whales and pinnipeds and the like. Following the megafaunal extinction in North America, condors' range was reduced to the point where they were most common along the coast of California, and they focused on a diet of marine mammal carrion because the megafauna had become extinct. In historic times, after the arrival of Europeans, they focused on range cattle and carrion of deer and feedlot cattle. The reintroduced populations, as a matter of fact, are supported largely by stillborn calves provided as carrion for the reintroduced condors. So the cool thing in here is that the point I want to make is that these condors were re-released, rewilded, if you will, into the plateau country of northern Arizona and southern Utah, by virtue of the fact that we knew they used to be there and we knew what they lived on in the past. They were also reintroduced to here in their range during historic times along the coast of California, and it's not clear to me whether there was evidence here in Baja California for pre-existing condor populations, but the habitat is pretty similar. So this is really Pleistocene rewilding in northern Arizona and providing them with a different diet that they than they previously knew. And besides this particular spectacular image of a condor along the course of the Colorado River, here are some cliffs along the Vermilion Cliffs in southern Utah. And while you can't really see any condors, you can see their excrement as indicated as to where these birds now nest. This is a case of intensive management with very, very high costs for both the captive breeding programs and re-release and feeding of these animals in the wild. Unfortunately, this particular species is now threatened by lead in the carrion of game animals that these birds now feed upon. So this is an ongoing story, but it's a highly charismatic species and a lot of money and effort is going into its preservation. And I want to switch to a much smaller bird, a much, much smaller bird, southwestern willow flycatcher, the third of my 3 stories. This is about the size of a sparrow, in case you want to get a grip on that. These are migratory birds. They nest in cotton willow riparian habitat in the southwestern United States, in the arid regions of the southwestern United States. This is a bit of riparian habitat of willows, and this is actually a nest. They make quite large nests. But the point here is that riparian habitat is disappearing in the Southwest due to the extraction of water by groundwater or diversion of surface waters. This is some more of its habitat in some restored sections of the Colorado River Delta. And these trees here, these are some willows. These are only about 6 years old, so they grow quite quickly when provided with ample supplies of water. There's less water because surface water is diverted for human uses, either in agriculture or for cities. And that water that does exist no longer follows its natural hydrograph. This is the hydrograph of the Colorado River before an enclosure at Glen Canyon Dam. The blue is before Glen Canyon Dam, and what you see here is a big spring snowmelt. At the present time, flow in the Colorado River is heavily managed to even out that flow throughout the year for purposes of providing water for agriculture and also for power generation from these dams. So it's a much flatter hydrograph at present than it was in the past. But what happened usually in the past were spring floods that serve to distribute the seeds of cottonwoods and willows and thus propagate the cottonwoods and willows. So it's this lack of seasonal flooding that hinders recruitment and constitutes habitat loss. You can get recovery and maintenance of these habitats, as in the case of this experimental flood, an engineered flood in 2014. But again, this is an investment of both water and effort into restoring and sustaining these particular habitats. That's the bad news. Habitat, the natural habitat of the southwestern willow flycatcher has been reduced. Willows and cottonwoods are now being replaced in a lot of riparian habitats in the Southwest by an invasive species, salt cedar or tamarisk. These were introduced into this species, several species were introduced in the late 19th century to serve as windbreaks and ornamental trees. They're drought tolerant, tolerates brackish water. So it's a very, very hardy species, but it's taken over in areas that no longer have extensive water, that no longer have seasonal flooding. It's a nasty little tree. However, the good news is, that this tree now provides nesting habitat for southwestern willow flycatcher. So the flycatcher says, okay, no more willows around, I'll settle for salt cedar, and it does settle for salt cedar. That's the good news. Now, two bits of bad news are that tamarisk creates a fire-prone habitat. It's a much drier species, and fire moves through these habitats rather quickly. The second bit of bad news is the introduction of the tamarisk leaf beetle, introduced as a biocontrol in 2001. And there you have the perpetrator here in the upper right. And what the tamarisk beetle does is it defoliates the tamarisk, exposing flycatcher nests to predators. Okay, so just because you've defoliated and reduced the distribution of tamarisks doesn't mean you automatically bring back the native habitat of cottonwoods and willows. That has to be done through active restoration and irrigation. So again, extensive effort to bring back the natural habitat of the southwestern willow flycatcher. So really, three generalizations from three species, I should say. Hard to know how to generalize from just three, but perhaps it might be useful to again note that endangered species policy typically focuses on habitats and preserving and restoring those habitats. In the examples I've cited, niche labelability at decadal timescales is a good thing. And I say decadal because that is typically the time period over which policy moves. At best, you've got 50-year commitments to restore habitats, but in typical timescales for funding from government sources or even NGO sources, we're talking decades. Third point of this that I hope I've made with the first two examples is that the fossil record can indeed be useful in bringing back some of these habitats and bringing back some of these species. And the final point throughout is that nature needs help. Active management is necessary in all of these cases, because after all, we don't just want to understand nature. The point is to defend and preserve it. Thank you for your attention.
Thank you very much, Carl. That was really, really fascinating. I'll call for any questions in the Q&A box. See if anybody's got some questions. Can I ask one while we're waiting, perhaps?
Of course.
Being interested in mollusks, I've gone back to the millennia. I'm really interested that the numbers have dropped because the salinity has gone up because the Colorado has stopped running. But you've clearly got this population somewhere which actually enjoys really rather high salinities. Do we know? Well, you know where it is clearly, but do you know how sort of derived is that? You know, how long have there been populations, do you think, that have been able to stand much higher salinities?
Yeah, you know, we paradoxically we concluded that Mulinia was a brackish water species before we had sort of found these remnant populations elsewhere. These populations that exist in high salinity areas, you know, those high salinity areas weren't present when the Colorado River was still flowing. But quite clearly, there's some tolerance for them. The interesting factor here is also perhaps that the Colorado River has been known under natural circumstances to switch its course, such that for a 20 or 40-year period of time, it would empty into the Salton Trough and thus cut off its natural flow all the way to the Gulf of California, thus causing an increase in salinity in those areas. Yet, quite clearly, populations of Mulinia bounced back when the river resumed its course. So I suspect there was always that potential for Mulinia to survive in relatively higher salinities than in the past.
Yeah. Okay. And how do they get on when you transplant them?
I'm sorry?
How do they get on if you transplant those ones to other parts, or have you not done that?
Haven't done that. That's wonderfully ambitious, and I'd love to be able to do that.
Sorry. So Andy Johnson's got his hand raised. Here we are.
Really, only as a follow-up to what Liz has asked there, really. I think what you're saying effectively there, Carl, is that it's actually changed its fundamental niche rather than its realized niche. Would that be a fair statement?
I don't think so, Andy. I'd say it always had that potential. I'm not sure that this was an evolutionary change. Yeah, gosh, that's a terribly academic question. I honestly don't know the answer to that one.
Fair enough.
Well, this is supposed to be a discussion meeting, so that's good. And actually, I see in the chat that Annarizko has just asked a question that I was flitting around my molluscan mind as well, in that these populations in higher salinities, how do they sort of compare in terms of sort of biomass, shell thickness, and so forth from those in the other populations?
I think from terms of, I was never able to harvest a live Mulinia when the river was still flowing. So I can't talk about, I wouldn't be able to compare soft tissue biomass, but in terms of the shell thickness and shell size, they are comparable. They're a bit touchy when you look at their sclerochronology to sort of get at their growth rate. They're not as easy to deal with as Venerids are, say. But I would say they're comparable. Yeah. They're comparable.
Okay, that's interesting. Any more questions? Right. Well, I think in that case, we probably move into the break at this stage. So thank you. That was a really interesting talk. I've never seen condors with numbers on their wings before, so I was excited by that. So we've got a 10-minute break now for everybody, and we'll start the next session. And I think Richard Twitchett should be along. He's here now. He'll be chairing that one. So thank you very much to all three speakers. It's been really fascinating. Thank you.
Is Becky there?
Hello.
Hello, Becky. Just a quick question for you. You may have heard me saying, it doesn't seem as though we can put questions in the question and answer box. There doesn't seem to be an...
Yeah, I did say that would happen.
Did you?
So,
yeah, I must have missed that. So we have to put them in the chat, whereas the normal delegates can use the question and answer. Yeah,
that's correct.
Yeah, understood. I know we're not supposed to be talking in this part of the proceedings, but I thought I'd better get that clear. Okay, then. Well, we're keeping very nicely to time.
Yes.
Well, just a minute or two ahead of the scheduled break, but that's very good, and it's working very smoothly so far.
Yeah, and some interaction, which is great. So thank you very much.
Yeah, that's good. No, Liz handled that very nicely as well. I don't know whether she's hearing me as I speak, but that seemed to go very well.
Okay, well, I'll see you after the break.
Yeah, all right then.
Okay, welcome back, everyone. I will resume the second part of the discussion seminar. So it's a great pleasure to welcome to the event my colleague in the Natural History Museum, Tom White, who will be talking to us about Pleistocene interglacials. How warm were they? Reconstructing past climates using terrestrial fossil assemblages. So over to you, Tom. Thanks.
Thanks, Richard. Sorry, I just want to unmute myself. And that's working now. I'm also going to hide the meeting controls, otherwise I tend to get in the way of what I can see on my screen. Yeah, thank you very much to Andy and to everyone who sort of put this meeting together. I think it was way back in the distant past of 2019 that Andy came to the museum to look at some bivalves in my collection, and we started talking about some of these things, and that's where the idea to sort of put this kind of meeting together started to gel. But it's been a difficult 2 years to get that sort of thing done. So I hope this is just an interesting jumping off point really for discussion, hopefully. So my background is in Pleistocene interglacials and Pleistocene paleoecological reconstructions. I'm a malacologist by training, but I sort of dabble with other groups as a part of this kind of multi-proxy reconstruction work. And I note that my colleague David Horn, who gets mentioned in this talk quite a bit, is here. So hello, David. And I just want to talk through really very broad and quite rapid, given I've only got 15 minutes, overview of quaternary paleoenvironmental reconstructions using sort of terrestrial fossil assemblages. And the main theme of the talk is going to be temperature reconstructions. And so it's a critical appraisal of how these fossil proxies can be used to reconstruct past environments, and how careful use of multi-proxy data sets can provide a more consistent and reliable set of evidence for past climate and environmental changes. Yeah, and also just my general aim is to generate discussion, as I said. I'd like to just also flag up some collaborators who've worked with me on various aspects of this over the last 10 years or so, including colleagues at the NHM, Ian Candy at Royal Holloway, David at Queen Mary, and Jonathan Holmes at UCL. So in case anybody here is not familiar with the recent geological period, the Pleistocene, and the way in which we generally talk about the environmental fluctuations that have occurred during that period. This is the marine oxygen isotope stage curve, which was the data set provided by Lizicki and Raymer in 2005, tends to be the basis for most of these diagrams. And here we are in the Holocene, which is actually stage 1, not written up here, and these repeated fluctuations between even-numbered and odd-numbered stages are essentially glacial episodes with high volumes of ice at the poles, and here in Northwest Europe, quite inhospitable period. This is the last glacial maximum during stage 2, and interglacial periods, which are the more the temperate periods between glaciations when the climate was conducive for occupation by all sorts of animals that prefer more temperate conditions, including humans, of course. And a lot of the work I've done over the years has had an archaeological component, and many of the questions asked are sort of why, what was the environment like when humans were populating parts of Britain or Northern Europe during these various interglacials? And that, of course, leads you to think about these uniformitarian concepts quite closely, because you think, well, how much information can I reliably give you from a group of broken up old mollusk shells about the climate and the environment that was being encountered by people and animals in the past? So I'm going to focus today on stage 5e, the last interglacial, which is about 125,000 years ago. In Britain, known as the Ipswichian interglacial, because the locality for the interglacial is a place called Bobbitt's Hole near Ipswich, and it stems from work that I did relatively recently with Ian Candy, really trying to critically appraise how warm that interglacial really was, and whether we could get any more detail from the fossil record in the UK. So this is a reconstruction of the British last interglacial as published by Tony Sutcliffe in the early 80s. And it gives this impression of it being an extremely warm, sort of balmy, almost sub-Saharan African paradise. So very famously, of course, it's known for having hippos in the British mammal fauna. But there's also lots of other things here that give a very distinctly African flavour: spotted hyenas, straight-tusked elephants that are extinct, and various other species roaming this paradise. And it's also famous for lots of other things in the invertebrate fauna and the botanical fauna, the tree fauna from the pollen. Personally, of course, I'm very interested in mollusks, and when it comes to reconstructing these environments, I tend to go for these shorthands for different places that you might see. So if I was to find woodland taxa in my fossil assemblage, I might conjure up an image of a woodland like this, a nice deciduous place, or perhaps I might find something with these hygrophilous species. And Rashid was saying earlier about some of these sort of lazy shorthands that kind of infect disciplines. And I think, Ian and I are interested in questioning some of these issues. To what extent are some of these ideas about past temperatures are sort of embedded in our understanding, entrenched in the field, and sort of lazily cited. So we find a certain taxon, whether it's a mollusk or a tree, and you think, okay, well, automatically that must infer warmth. So there are various other more detailed methods of getting at temperature. There are the mutual records, the MCRs and the motors, using beetles and ostracods, which you can use based on populations of different species. You can obtain further constraints from species known to have particular thresholds above or below which they can or can't survive, or perhaps influencing when they can fruit or when they can reproduce. It tends to be the case that cold thresholds are a bit more difficult to quantify with precision. So the emphasis has always really been on summer temperatures. But basically, by combining the temperature requirements of multiple species using mutual methods and using individual taxa, the winter and summer temperatures can be more robustly constrained when producing a reconstruction for a given interglacial. And just mentioning pollen here, I'm not using pollen in the temperature reconstructions per se, but it is a very valuable means of locating the assemblages within an interglacial. So Rashid mentioned and showed some diagrams showing pollen reconstructions across interglacials, and these are the things that will allow you to sort of locate your sequences and tie them together, because most of the time we're way beyond the range of absolute dating methods such as radiocarbon dating. It's not clicking. There we go. So I'll just quickly outline the mutual range methods. Possibly the most long established are the MCRs based on beetles, but that sort of method's also been applied to herpetiles, vertebrates. And Dave Horn developed the mutual ostracod temperature range method using non-marine ostracods. Both of these methods use suites of species with known modern temperature ranges, which are based on current distributions, of course. But it does mean that necessarily you have to exclude extinct species or those with poorly known distributions. And sometimes, if based on the vagaries of the fossil record, you might end up with a rather limited fauna that you can actually use for these methods, because there aren't enough of them that are calibrated or have a known temperature range. But nonetheless, these methods find the mutual temperature range within which all of those species present could have coexisted. And here's an example nicked from Dave, so apologies Dave, but this is an ostracod assemblage, I think from the stage 9 site, and basically the blue box shows you that temperature range within which all of those known species or extant species could have existed in the past. But note that any value within that range could represent the true mean past temperature. So there isn't any value within that range between -4 and +1 degrees that is the correct answer, as it were. So there are some difficulties inherent in this. One is that critical appraisal of this evidence requires detailed knowledge of the current ecological constraints of the species you're finding in the fossil assemblages. And often, especially in the case of invertebrates, there just isn't enough information from any species. We don't know. You ask basic questions like, how tolerant are certain bivalve species of salinity levels, and we just don't know. So one of those issues is that there isn't enough information. Secondly, that modern ecological studies tend to focus on invasive species, especially plants. And so, run-of-the-mill species that we might find regularly in our Pleistocene assemblages just don't really get much attention. And also that modern distributions aren't necessarily a reliable indication of species tolerances whatsoever. So going back to the example of hippopotamus, it's now sort of a well-known sub-Saharan African mammal, but that's only been its distribution and range relatively recently. It was until the beginning of the Holocene, essentially a circum-Mediterranean species. It's all over southern Europe. It was only extirpated from northern Africa, places like Egypt, about 4,000 years ago. So to sort of call hippopotamus a sub-Saharan species and infer it must prefer very warm temperatures is completely wrong. And similarly, there were formerly widespread species that are now very, very restricted. So there's a species of hydrobiid mollusk, gastropod called Belgrandia marginata, which is a really common component of nearly all of the post-Anglian Pleistocene faunas across Europe. And yet its modern distribution is restricted to some warm freshwater springs in southern France. And so the question you have to ask is, well, was Belgrandia in the Pleistocene somehow driven back to these final outposts in these springs in France? I don't know by what kind of environmental change or catastrophe that would have affected it when it was so widespread previously. Or is what we're looking at in our Pleistocene fossil assemblages very similar to Belgrandia marginata, but actually a different taxon that we can't really identify based on shell characters alone? So there's a question for the discussion. And also when you're looking at colder environments, which tend to be the lesser well known, there just isn't very much information about tolerances and thresholds for cryophile species versus thermophile species. And there are also some inherent dangers in using data sets based on literature and on museum material, fundamentally misidentification and inconsistent identification of species. So that's very important to check if you can go back and look at material that's been published and find material in museum collections to make sure that you are indeed looking at the correct taxon. Sometimes with the neutral ranges, if there's an over-reliance on a single taxon to produce a tightly constrained range, you have to perhaps ask whether that is giving you a biased result. And also very careful consideration of where material came from in complex sedimentary sequences to make sure that stratigraphy and taxonomy are really well sorted out so that there aren't mistakes. So beyond the mutual methods, we can also derive thresholds, temperature thresholds from known ecological preferences of certain taxa. So here are some quite well-known examples of species that have, well, particularly these thermophiles, minimum constraints, minimum requirements for mean summer temperatures. So we have Emys ubicularis, which is the European pond tortoise, which really needs temperatures above 18 degrees to successfully reproduce, and even higher than that, really, because it has temperature-dependent component to its eggs, so that if they are not incubated at a certain temperature, you won't get a sexually dimorphic population that can reproduce. It'll be either all males or all females. And then a few plants here, Salvinia natans, Nalus minor, and then famously Trapa natans, the water chestnut, which provides this kind of very high temperature, minimum temperature requirement of 20 degrees mean temperature for summer, which is supposed to be the minimum temperature below which it won't successfully fruit. And there's also sort of simple things like adding in deciduous woodland ecosystems need something around about, well, -2 degrees is the minimum winter temperatures, any lower than that, we won't go to deciduous woodlands. So if you're getting a woodland pollen signature, you can infer that those winter temperatures might be higher than that. But again, there are complicating factors. You know, we're trying to reconstruct temperature, but temperature may not be the only variable operating. So there are issues like insulation. So sometimes, as I was saying, Traponatans, which is a floating macrophyte, can survive in lakes at high latitudes, sometimes in areas which are, on paper not warm enough to support it. And individual microclimates can also come into effect. So with MS, usually a southern European species, but there are outlying populations in Germany which live in south-facing slopes that maintain temperatures where they can actually reproduce. And statistical considerations, and just a number of taxa are available. So we want to make sure that there's a good number of species thrown into the mix to make sure we aren't basing it on just one taxon. So finally, having just whistled through all that information, I'd just like to show the data we assembled for the last interglacial climate in the UK, in Britain. And partly one of our motivations for doing this was to test the idea that the last interglacial, the Ipswichian, was a partner for future climate change. So, could we discern any patterns in the climatic changes at these various sites in the UK, showing the map here, that would corroborate or not the similarity between the last interglacial and the potential progress of the Holocene. And here, this diagram sort of brings together all the things I was mentioning into one diagram. So these are the sites from which we've derived a variety of different paleo-environmental data. We have MCR data based on beetles, showing the blue boxes are the ranges for winter temperatures, and the pink boxes are the ranges for summer temperatures. We've ordered them roughly by their position relative to the Ipswichian last interglacial pollen record. So the sites below this line are earlier in the sequence than the sites above. And the two grey bars are the modern mean temperatures for winter and summer. So here we have mean winter temperatures around about between 3 and 4 degrees, and summer between 16 and 17 degrees. And then these other proxies sort of mapped on here to show you the requirements for temperature that have been shown by the proxies found in the assemblages. So essentially, in this early part of the record, the controlling factor is MS orbicularis, with its sort of plus 18 or 18 degrees mean temperature requirement for the summer, which is slightly higher than modern summer temperatures. Not a lot different here in these sites, but then as we get into these localities, we see that things like Traponatans, with its nominal 20-degree minimum temperature requirement, is really driving an increased summer temperature record for the Ipswichian at Trafalgar Square. But when we compare these records with the Holocene, it becomes clear that there are several issues. One is that the Thermophilus species that are common in these 5E sequences are absent or very rare in the Holocene. So there clearly is a major difference between the two interglacials. Secondly, that the heightened temperature at Trafalgar Square is only due to a single taxon. So although Traponatans gives you this plus 20 degrees theoretical minimum for that site, in actual fact, the other mutual ranges and other species present suggest something possibly slightly cooler. And in fact, as we found, the pattern of increased warmth is not consistent with predicted Holocene temperature increases, particular winter temperatures. So as I hadn't really mentioned this, but the winter temperatures are all consistently much colder than the modern averages. And so there's a much more continental, much more seasonal flavour to the environment of the last interglacial. So although the climate of Britain during 5E, during the Ipswichian interglacial, was warmer than present day, only one locality, Trafalgar Square, indicates a degree of warmth that's greater than the Holocene thermal maximum. And it's not really analogous to future climate predictions for Earth from 2100 onwards. And finally, just to show you that when you apply this same sort of methodology to other interglacials, going back in the British record through the late Middle Pleistocene and the early Middle Pleistocene, we get a very similar flavour in each one. And that's primarily because we have a sort of recurring pattern of species that repetitively turn up in our Pleistocene fossil faunas. So we get a lot of MS, we get a lot of Trapa. They turn up in all sorts of different time periods, going back to sort of MIS 15 here, MIS 9, MIS 11. The same taxa are turning up. So the climate appears to be more continental and more seasonal during present day and during the interglacials than the Holocene in the present day. And the structure can be shown to be sort of very similar across essentially well over 700,000 years of Pleistocene time. So going back to the Ipswichian and how we think it might have looked, I'll say thank you very much for listening, and have you got any questions?
Thanks, Tom. That was a really good talk. So let's have a look at the questions in the question chat. I think we've had a few going through here. So we have a question about hippos. Is the Holocene reduction in the range of hippos a function of human impact? And we had that's from Andy Johnson, and Malcolm Evans replied that he's unaware of a single hippo subfossil from an archaeological assemblage Holocene in Europe. So do we think it's human impact? Is that behind the hippo story, Tom? I realise it's outside your mollusks.
It's outside my mollusk, but I did do quite a bit of work on hippos. I did some work in Arabia a few years ago. As far as I know, there's evidence of human predation of hippos in places like Egypt, because they were common in North Africa until relatively recently, and I think it's supposed to be linked to human impact there, that they were driven back to the sub-Saharan part of Africa. But prior to that, I don't think there's any evidence of human impacts affecting them. It's probably a climatic change that drove populations back out of Europe and around the Mediterranean.
Great. Okay. Cheers. There's a comment from Dave Horn. Excellent talk. Nice slides. And you're rightly cautioning against too much reliance on a single taxon, a single beetle or ostracod to give a tightly constrained mutual temperature range. But is that really any different from using single indicator taxa such as Emmys or Bicularis to determine threshold temperatures?
No, and one of the things was that we were trying to get to with our work was to what extent can you rely on those nominal values ascribed to things like Emmys and Trapa. As Rashid was saying, there's a laziness involved in some of these things. You just throw them into your analysis because they've occurred, and this is what's been told. This is what's been published prior. Yeah, I think the issue is that we can never really disentangle some of the different variables that are operating, and so especially with the difficulties interpreting fossil assemblages and all the taphonomic factors that affect those, and I'm not sure we'll ever know. But yeah, I mean, we can talk about this a lot, Dave, because we've as we have done. But yeah, there's probably not much difference, to be honest. Okay,
and we're a little bit pushed for time, but quick answer if you've got one there, Tom. From a question from Jess McCoy. Apart from comparing the tolerance ranges of species, have you looked at other methods of reconstructing the paleoclimate tolerances at all? So presumably chemistry or something like that, I guess.
Yeah, I mean, so my colleague Ian Candy at Royal Holloway is doing a lot of work on geochemistry, using things like biomineral from mollusk shells to try and get at the temperatures directly. So using oxygen isotopes. I think there are issues in trying to get a direct link between the measurements you might take from the biomineral and actual air temperature, which is what we're trying to reconstruct. But yeah, I mean, there are other aspects of paleoclimate that you could think about using these things. I mean, salinity has been mentioned already, and from a point of view of Britain and its fluctuating island and peninsular status of northwestern Europe, and sea level, which is inherently bound to climate change and fluctuating ice sheets. Yeah, we could look at that as well. So yeah, there are lots of other things that there's too many to really discuss in a 15-minute talk. But hopefully, if we get to do a bigger version of this meeting down the line, we can talk about them then.
Indeed, absolutely. Thank you very much, Tom. That was great. So we move on now to talk from Jan Hennison from the British Geological Survey, who will be talking to us about the late Neogene to early Quaternary seasonality changes recorded in North Atlantic dinoflagellate cyst assemblages. Down there.
I hope you can see my screen now. That you can see the presentation now. Okay. So thank you for inviting me, Andy, and I hope that one day soon we can have a coffee again at the BGS. I'm a senior palynologist and the biostratigraphy and paleontology lab manager at the BGS. And today I'll be talking about how seasonality changes have been recorded in dinoflagellate cyst assemblages at the Pliocene-Pleistocene boundary, and how niche stability of dinocysts can be tested and employed to evaluate climate dynamics at that boundary. So at the top left-hand side, you see an overview of the thermohaline circulation, and it indicates how important the North Atlantic is in that diagram here. So you have two yellow dots there indicating where deep water is formed. I'll just switch to a laser pointer. And deep water travels all the way down to join the Antarctic circumpolar current, and to maintain mass and energy balance, a superficial stream of warm superficial waters flows back into the Atlantic. So that's why the Atlantic is such an important area in this diagram. The interval that I will be talking about is straddling the Pliocene-Pleistocene boundary here, this is around 2.6 million years ago. And this is the same diagram that Tom used in the previous talk, indicating the changes throughout this time in history. So you have very warm, relatively ice-free conditions in the Pliocene, and all of a sudden around the Pleistocene, you see this sort of nosedive where we go into the Quaternary style ice ages that Thomas also has talked about. And the interval I'll be talking about is indicated here in the gray box. So why is the Pleistocene climatic transition so important? So first time I gave a similar talk on this topic was in September 2013 for the annual meeting of the Palynological Society, and I just pulled some data of the National Oceanic and Atmospheric Administration website, where they measure the CO2 in the atmosphere at the Mauna Loa Observatory. And that was the first time around that time that we surpassed the 400 parts per million. And now I did the same again last week when I was preparing for this talk. And you see that now we've already moved up way beyond that. And I expect if in the 10 years or so, I give a similar talk, I will be sitting over here somewhere. But how is this relevant to the Pleistocene, or the late Pliocene at least? On the right-hand side now, you see a compilation of all data that I could find about partial pressure of CO2 in the atmosphere around that time. And this is expressed in micro atmosphere, which is 10 to the minus 6, which is comparable to parts per million. So broadly, we can, for comparison reasons, we can draw this line over here. And then you can see that the late Pliocene, early Pleistocene is the last period in Earth history where we have a sustained period where there is CO2 in the atmosphere that's comparable to what we're experiencing right now. So it is an important time frame as a sort of paradigm for what's going on in climate right now. Why then look at the Pleistocene of the North Atlantic? Well, the map that you see on the left-hand side here was produced by people who were working at the PRISM group and looked at the Piacenzian, which is the last part of the Pliocene, period from 3.29 to 2.97 million years ago. And they used an array of proxies, and they compiled this map over here, which shows you the temperature difference between the Pliocene oceans and the extant oceans. And the darker the colors are in this map, the higher the difference is. And you can see that some of the darkest colors occur here in the North Atlantic. So clearly the North Atlantic was very, the changes that occurred between now and the North Atlantic in the Pliocene are very, very big, and some of the largest that have been recorded. So for the rest of the talk, I'll be focusing on samples that I took from 2 sites in the North Atlantic. So the map on the right-hand side, you see here, the colors here are expressing the sea surface temperatures that are pulled from the World Ocean Atlas. And superimposed on that is the superficial currents of the North Atlantic in the gray here. So we have the Gulf Stream and the North Atlantic. And the samples that I will be talking about are originating from Site 610, straight in the path of the North Atlantic currently. And U1313C, which is here in the subtropical gyre just to the south of it. So why would you apply palynology to this equation? Why would you use palynology? So on the left-hand side, you see image of that I captured of Operculodinium centrocarpum sensu-Wallendale, which is a dinoflagellate cyst. So this is the cyst stage of the life cycle of dinoflagellates. This is what we find in the fossil record. And this is the same as another part of the life cycle of the same taxon is this motile cell called Protoceratium reticulatum. But in the fossil record, we only retrieve the cyst, of course. And in 1983, a colleague of mine at the British Geological Survey, Rex Harland, produced these maps, which is basically a compilation of 142 sites where he took bottom samples and then did palynological processing and counted all of the dinoflagellate cysts that were encountered and did a relative abundance analysis, basically. And the stipples that you see here, the lightly stippled areas, is where Operculodinium centrocarpum occurred between 0 and 10%. In the middle dark here is the abundance was between 10 and 50%, and the highest abundances were recorded in the darker areas where they were dominant over 50%. And I think you can agree with me that the currents that are displayed here in the little map on the bottom are mimicked fairly well, especially the North Atlantic current, by the abundance profile of Operculodinium centrocarpum. And since then, it's been used as a robust indicator of the North Atlantic current, at least the distribution pattern of Operculodinium centrocarpum. I'll briefly go over the methodology that I used to look at the Pliocene samples now, because it bears significance for what I'm going to be talking about later on. So you collect the samples from the IODP repository in Bremen, you try and weigh them, disaggregate them, and then the most important step occurs here, where we wet sieve our samples on a 125 micron mesh. This means that we can effectively separate all of the foraminifera and the palynological material, or the material that we will be using for palynological processing, because that is smaller than 125 microns. So we have the forams we can use for geochemistry, for oxygen isotope analysis, for delta 13C, and also for magnesium calcium, which will be the basis of our sea surface temperature reconstructions. And then the dried filtrate, that's the material that went through the sieve, we conduct palynological processing on, which is basically pouring a lot of hydrochloric and hydrofluoric acid on it and extracting the organic matter and putting it on a microslide, looking under the microscope and do the counts. So we have two independently derived, independently derived environmental factors that we then can relate directly to the changes in the dinoflagellate cyst assemblages. And we can bring them back together quite easily because they are originating from the same sample. So we have the same sample multi-proxy approach. And here is a snapshot of some of the results that you can gain with an approach like this. At the top of the diagram here, you see the temperature record of U1313C, which is the more southern side displayed here. Below that, see the temperature of 610A in black. And beneath that is an overview of the dinoflagellate cyst assemblages. So each color in this diagram represents a single taxon of dinoflagellate cysts. And the top one here is for 610A, the site here, and the bottom one here is for U1313C. So the first thing to note is that there was a major drop in temperature in 610, around 2.72 million years ago. And you can see that, I should mention, the yellow in here is our Opercular central carbon sensor Wallendale. So that is our indicator of the North Atlantic current. So at the first drop of the temperature here, you can still see that this cyst is still very much occurring in 610. While over here, if we move on to marine isotope stage 4, which is at the very base, or at the transition from the Pliocene to the Pleistocene, you see that this completely disappears out of the record. And it is replaced by this purple color over here, which is Pyxidonopsis braboi. This is a fairly poorly understood species, or is fairly, it's not widely recovered. And the times that it has been recovered was, for example, in very extreme conditions in areas where there was a lot of ice cover. So this is clearly an indicator, or if we apply the ecological uniformitarianism principle to it, if that rings true, is clear an indicator for cold water conditions. So what we proposed is that this material, or the material that contained Pyxidonopsis braboi, came from further northern latitudes and was present very much at this time over site 610A. Opercular central carbon was recovered in its highest abundances further south at that same moment. Further to note as well is that at this point, at this, the same marine isotope stage, you see a collapse of the meridional sea surface temperature contrast. So basically, you were recording temperatures at U1313C that were similar to 610A, even though U1313C is significantly further south in the same oceanic area. So what could explain these changes? What I already explained that the North Atlantic current and a lot of the currents there are forced by one forcing mechanism, the thermohaline forcing. That was in the first slide, and I briefly explained that. But there is another component to it, and that's the wind forcing. So in the modern conditions, the wind forces the North Atlantic current in the mid-latitudes, forces it to go to this side. So if you want to suppress that, these strong westerlies in the mid-latitudes, two things need to be present. You need to have increased atmospheric pressure in the high latitudes here. So at the same point that I recorded this shift of the North Atlantic current, that's around the same time that the entirety of Greenland is supposed to be glaciated. And over ice sheets, it's normal to have anticyclones, which could establish like a high pressure area. So it's a hypothesis that this could have been the case. But one form of firm evidence that we have is if you want to reduce your westerlies in the mid-latitudes, you need to have a reduced meridional sea surface temperature gradient. And that's exactly what we observe. It's plotted here again. So at this marine isotope stage 104, at the very base, at the transition between Pliocene and Pleistocene, you see this collapse of meridional sea surface temperature gradient. And then I did something that, paleoceanographers should probably never do, and is compare your results to other researchers. I noticed that there were two other studies that did research on the same exact borehole, site U1313C. And I plotted their results next to my results, and it's displayed in this diagram at the bottom here. So my results are in black here. And then David Navs in 2010 published a similar record in green here. And then at the top you see in red, the sea surface temperature by Friedrich et al. In 2013. And so I started to wonder, why are these so divergent? And then I realized that we all three used a different biotic carrier, and that's a very important thing. So the red diagram is constructed using this foraminifera here, Globigerinoides ruber. And my diagram here, the black one, was used with Globigerina beloides as the biotic carrier, while David Navs used alkanones, which are the organic lining of coccolithophores. So that, how could I explain then the difference that we see in temperature? So I looked at the current ecology and the niches of those three taxa in the North Atlantic. So Globigerina beloides and ruber both occupy surface waters, the top 60 meters. So they should record the same sort of depth range. However, I found that nowadays, Globigerina beloides is most abundant during early spring, while Globigerinoides ruber seems to be most abundant in late summer, in August. So I hypothesized maybe we could use this as a paleoseasonality indicator if we subtract the two, as a very crude tool for paleoseasonality. At the same token, alkanones are produced by coccolithophores, and they have two blooming seasons in the North Atlantic. So they bloom in the spring and in the autumn, and that explains why they have it approximates more an annual average sea surface temperature, and that explains why the green record that you saw in the previous slide is mostly in between the other two. So if we use the principle of niche constancy and we assume that the blooming seasons were very, very similar, then we can use this as a paleosalinity or paleoseasonality indicator. Sorry, I keep on saying salinity, it's paleoseasonality. And that is plotted at the bottom here. And that paleoseasonality indicator peaks at the exact same moment that the full glaciation of Greenland is hypothesized. And that at the same moment that we have a southern shift of the North Atlantic current that I showed to you earlier. So it seems that this may be a plausible seasonality indicator. And one more thing that I want to highlight here, the technique that we used by separating out the foraminifers and the dinoflagellate cysts, we can also evaluate some niche constancy between the Pliocene or in the early Pleistocene and the extant dinoflagellates. So for example, at the left you see an image of Spiniferitis mirabilis, a very well-known dinoflagellate cyst and always associated with warmer waters. In the middle diagram, you see a diagram composed by colleagues of mine who also looked at Pliocene samples and early Pleistocene samples, which are displayed here in the brighter colors. And then in the background, you see the gray colors, and these are the results of an extant analyses of the Northern Hemisphere bottom samples. So this is also the relative abundances of the same species, but then for extant samples. And you see that the Pliocene samples and the extant samples have an excellent fit, and it's almost a nice unimodal distribution with the ecological optimum around 15 to 16 degrees. And the results that I obtained were very similar. So the same species recorded in my samples was also 14 to 16 degrees where it reached its highest relative abundance. However, we also uncovered a few others which shows that there is niche lability, as people have called it earlier in the talks. And the best example that I have for this is a species called Impagadinium pallidum, which in the extant catalog it's always been used as an indicator of very, very cold water, so even temperatures below zero. So they actually appear in the very high north latitudes, and that's where they reach their highest relative abundances. However, in the Pliocene, this is a completely different story, and we found that their optimum seems to be between 10 and 15 degrees. It's the same that I found here as well. So the optimum for this species, for this taxon, is in much higher temperatures. There are differences in at least in sea surface temperature optima for these different species. So this is a way, by using these independently derived environmental factors and then apply them to the palynological record, that we can show whether there is niche constancy or there are changes in the niches of some of the species. So a few conclusions of the work that I've done. So palynology, we use an assumption of niche constancy to allow the interpretation of certain paleoclimatic and paleoceanographic changes with some caveats. You saw the example of Operculodinium centrocarpum being used widely as an indicator of the North Atlantic current. But there are also ways of applying or looking at niche constancy across larger timescales by using these independently derived factors. And the example of Impagadinium pallidum shows that this species was warm tolerant when you go back to the Pliocene. One more thing that I didn't have really a chance to talk about, but which is relevant in today's context, is that this is again MIS 104 at the transition between the Pliocene and the Pleistocene. And in black here, you see the incorporation of dinoflagellate cyst in bottom sediments. And you see that in all of these Pliocene marine isotope stages, they are very, very high. However, at the transition here, when seasonality is the highest, you saw that the North Atlantic current went further south and was less strong. And we see that there's a lot lower abundance or a lot lower concentration of dinoflagellate cyst in the sediments. And because they are producing, together with diatoms, over 50% of our annual oxygen budget, this may have severe consequences. And the reason why I say this is that last year was reported in Science, in Nature Geoscience, that there is actually, that this slowdown is happening under the same CO2 conditions that we've now seen in the Pliocene. So, and that was a few conclusions on the work that I've done on dinoflagellate cysts in the North Atlantic. I'll happily take some questions.
Yeah, thanks, Yang. That was great stuff. I'm not sure we've got any coming through yet for the talk. Oh yes, we have. Jess McCoy just pinged one through. Out of interest, have you applied similar techniques to a different time period?
No, for this, the reason why we did that, as I explained, was because of the similarities in the CO2 composition that we have now. And I have not tried the similar techniques to different time scales. We should probably go further back into the Holocene, the Quaternary, actually, because we are bound to do these sorts of niche stability tests by species that are still living today. So Spiniferides mirabilis is one of them. Impagidinium pallidum is one of them, but we are bound by that. So that's a constraint that we have to do that. So we could come back closer to now and test that, but we can't go much further back in time, I'm afraid.
Yeah, I was going to ask something similar, just with that difference you pointed out with the alkanones and the data from forams, because presumably it's a general broad thing that when the seasonality of these blooms may explain actually some of the deeper time differences that we see between proxies, even though we don't have, you know, the living species there, I guess. Exactly.
So for a lot of the temperature records of this time frame, at least, those are actually extant species. So we could, it hinges on that assumption, which a lot of people take for granted, that those niches are stable and that they are behaving in the same way in the Pliocene as they were right now. So there's some work to be done to test this further, that's for sure.
Yeah, that's great. Okay, how are we doing? Got any more questions coming through from the audience? I can't see any at the moment.
Richard, there's one in the chat box that came through from YouTube.
Ah, is that for the previous talk there, Becky?
It could be, I'm not sure.
It looks like 5E, which is different time scale.
So, okay, Jan, thanks very much for that. That was great, really interesting there. I think as you say, the Pliocene is that last time period that we had the CO2 above 400, and I use that when I'm doing outreach events and things, talking about generation 400, those current humans who are only ever breathed more than 400.
Yeah,
Pliocene air.
We're going to have to find a different time frame very soon, because we're going to run out of that similarity is going to be gone in a few decades time, I'm sure.
That's very true. Yeah, excellent. Thank you very much. Let's close that down. Find the last talk now. So the final talk of the session today is Marcia Robinson from the US Geological Survey, Ecological Uniformitarianism, a Deep Time Perspective.
Great, thank you. Let's see. I thought I had this all. I'm sorry. Oh no. Sorry, I'm having trouble sharing my screen. I'll start in just a second. I do want to thank Jan for setting me up pretty well. let's see. Can you see my screen now? Hello?
No, we can't, I'm afraid.
Okay. Let's see. You know, Becky, I sent this to you in case something like this happened. I'm afraid it's because I've got a firewall on this government computer that sometimes happens in
I
some.
see. Give me a minute. Everyone talk amongst yourselves. Yeah, it looks to me like it should be working, but it's clearly not. So while we're doing that, though, first of all, thank you, Richard, for setting that up. And thank you to Becky for making this all work, and Andy for putting it together. I'm going to talk about work that we started about 30 years ago. Well, work that was started at USGS about 30 years ago, looking at planktic forams from the Pliocene. It's going to be a little bit different from previous talks, in part because of the age. It's clearly older. It's from about 3 million years ago. But also, whereas the other talks really, especially the earlier ones, really featured looking at studies that were featured at looking at niche stability. Our project, when we started, we just assumed niche stability and went forward. And now we are going back and looking at that assumption. And Jan talked about PRISM. I'm going to be talking about PRISM work. So that's the Pliocene Research, Interpretation, and Synoptic Mapping work. And this project was started in the late 1980s, and Harry Dowsett, my co-author, was there from the beginning, and I came later. And we were looking at Pliocene, this particular period of time, the 3.3 to 3.0 million years ago. For a couple of reasons, and that was to better understand what the climate was like during this most recent warm period, similar to today in some ways. And again, Jan talked about that, but also to help improve the climate models by giving them a past climate reconstruction that they could use to test their models. Becky, how are we doing? Can you see it?
No, I can't.
Can everyone else see it?
Yeah, we can see the title screen there, Becky and Marcy.
Oh,
Do you want
okay.
to go on to the next slide?
Yeah, I can't do anything, so I'm glad you can see it. I can't see anything.
1 minute. So it's the introduction page.
All right. So if you go to the next slide, oh, you've got the introduction. Okay, good. All right. So yeah, so we're talking the PRISM project. And so what we're talking about is Pliocene sea surface temperature, and that's what we use the planktic forams to do. And over the past 30 years, we've amassed a really large quantity of planktic forams from this time period. And we've used them in several different ways to calculate sea surface temperature, but all of them assumed species temperature tolerances haven't changed over time. Okay, next slide. So here's a really simple graphic that just sort of shows how SST estimation works when using species assemblages. So if planktic, well, we know that planktic foram abundance is primarily a function of temperature, but also a few other things. And if we assume those other factors are negligible, we can relate the assemblages to sea surface temperature, and we can take that back in time. So in this graphic,
If you look at time 1, for example, and the cooler species of foram are in blue and the warmer species are in red, you can see that in time 1 it's a cooler temperature because there are more blue ones. In time 2, it's warmer temperature because there are more red ones. And this has been done throughout the Pleistocene, and we've taken it back to the Pliocene. And in doing so, we assume that temperature doesn't change, or the temperature preferences have not changed. So that's the topic of this discussion, right? And it's especially important when we're going back 3 million years. Okay, next slide. So this is a statement that we made in 2007, that tolerances most likely drift with time. And we recognize that we're pushing things when we're using these analog techniques, but it didn't stop us at all. So next slide. By 1991, the PRISM project had reconstructed the sea surface temperature in the North Atlantic, and that's what's shown there on the left, and those were the sites that we had. And then by 2012, we had over 100 different sites, both IODP and, well, DSDP and ODP sites, and coastal plain sequences. And then the 10 years since PRISM, we've added a lot more sites in the Indian Ocean and to the North Atlantic. And if you could click for me. And all of our information from PRISM is used in data model analyses and model comparisons by the Pliomip group, which is the Pliocene model intercomparison project. And this chart shows how Pliocene model, the model sea surface temperature results compare to our sea surface temperature results. And these are the same models that are used for future projections. So it's important to look at whether our assumptions of niche stability is accurate, since all this data is in use. Next slide, please. And maybe it is okay, because in this recent study by Antel et al., coupled ocean atmosphere models were used to compare temperature and plankton foram abundance across a range of glacial and interglacial cycles. And they found that species occupied the same temperature conditions regardless of the magnitude of temperature change. So this is very good news for us. So maybe we're okay. And the specimen figured here, Neoglobochondrina pachyderma, I just wanted to mention that we would call that Neoglobochondrina incompta, and this is an extant Pliocene species, and this is a species I'll be talking about in a few minutes. So it's the same species. All right, next slide. So are our assumptions valid? Are the species temperature preferences static over 3 million years? Well, now we have a lot of new Alkanone generated temperature estimates. They're available in a lot of places, but many of them are available at sites where we also have our faunal assemblage data. And pairing these Alkanone temperatures that are an independent temperature measure to our species assemblages allows us to test these assumptions. So the first test that we did, which is on the right here, we were checking to see if species abundances are tied to SST. And so what's plotted here is all of our PRISM interval species abundances in the North Atlantic, against both the fossil, modern analog from planktiform sea surface temperatures, and those are the open circles, and the Alkanone sea surface temperatures, and those are the solid circles. And not only is this show that there is an overwhelmingly strong signal between temperature and the assemblage composition, but also there's some pretty good agreement between the temperature estimates from our fossil assemblages and the temperature estimates from Alkanones. So this is great. So then the next thing we did was to plot percent abundance of species in the PRISM data set to only the Alkanone-based sea surface temperatures from PRISM sites. And we compared these plots to modern core top abundances from CliMap and modern sea surface temperatures from NOAA. And I'm going to show that in a minute. But first, all Pliocene species are not extant. So next slide, please. So because of this, analog work that we've done requires grouping. So here we show the Pliocene taxa on the top and the modern core top taxa on the bottom that we grouped them with. The percent abundance is on the x-axis, and the sea surface temperature estimates are on the y-axis. So we've got three situations when we're working with the Pliocene and modern data. So click, please. First, the taxa in the core top calibration. There are taxa in the core top calibration that have evolved since the Pliocene. And when we have this situation, we group the Pliocene taxa with the next nearest living relative. So puncticulata and inflata and its descendant, inflata, are a good example of this. Next slide, please. And some Pliocene taxa have gone extinct with no modern descendant. Altispira is common in tropical Pliocene settings, but it went extinct about 3 million years ago. And we have grouped this with Globigerinoides seculifer because these two have the same biogeographic distributions in the Pliocene. Okay, next slide, please. On the far right, we have a sinistrally coiled Neoglobochondrina atlantica, which is the cold end member of the Neoglobochondrinids during the Pliocene. And then on the bottom is Pachyderma, which is a present day cold end member. Both of these are present in Pliocene samples, and we group the two, but because if you can see from those vertical dashed lines, the max abundance of Atlantica has a warmer limit, about 15 degrees, than the max abundance of Pachy, which is about 5 degrees. Grouping them together probably causes some of our samples with lots of Atlantica in them to have cooler sea surface temperature estimates. Okay, next slide. So most relevant to today's discussion is Neoglobochondrina incompta. So it's an extant species. It's in our Pliocene and it's in the modern. And we're going to talk about that in just a second. Okay, next slide. All right, so now I'm going to focus just on the North Atlantic and just on the sites where we have both our faunal assemblage data and alkanone data at the same sites. This is also our highest geographic concentration of data, and it's our highest temporal resolution data. So this allows us to do a focused comparison of our Pliocene species abundances to this independently calculated alkanone sea surface temperature. All right, next slide. All right, so I'm showing you the Neoglobochondrina incompta. So from the Pliocene is on the right, and the core top samples are on the left. And these distributions appear similar, right? So unfortunately, we don't have any final data from North Atlantic sites with alkanone estimates lower than 14 degrees. And this is because the Pliocene was warm. We hope to include data from 642 off Norway that we actually just received, but not in time to get it into this presentation, which will give us some cooler samples too. We did also include one site from the Western Equatorial Pacific, and that's 1115. So that's the light blue, to add some warm data at the top of the alkanone calibration. Okay, next slide. So these are comparisons to our extinct taxa, and they look pretty good too. The one on top, inflata to puncticulata, you can see in that area with the dotted lines around it that there's something going on there. And those data are from sites 951 and 958, which are circled on the map over to the right. These samples are responding to something other than temperature, but we don't really know what that is. It could be productivity or high nutrients. We're going to get more at this. This is probably telling us something really important, but we don't know what yet. Looking at the lower panel where we have seculifer and altispira, things look pretty good there too. The Pliocene altispira are plotted at the upper limit of the alkanone calibration, which is like 28.5 to 29 degrees, which means they're almost completely saturated. So that's as high a temperature as we can get from the alkanone estimates. Again, that's from site 1115 in the Western Equatorial Pacific. So where it was hot, it might have been over that. They might have been hotter than this. We don't know. All right, next slide, please. All right, so the real question is our documented similarity between the temperature responses of the Pliocene and modern Planktiform species good enough to do what we've been doing? So is it good enough to assume ecological uniformity over the last 3 million years and to continue this analog-based work? So that's the question. So this is the discussion part. This bucket of fossils to the left is from one of our field sites in Virginia. And I'm using it as an example here because if you look at that bucket of samples, you can immediately say that they were from a marine environment, right? And then based on the species that you see there, you can say something about their water depth, maybe something about the temperature, maybe something like the water must have been clear and low energy because some of these are filter feeders. And my point is all of these are modern analogs, right? These are all analog interpretations and analogs to modern species tolerances and their environments. And we can continue with these interpretations until we can maybe even assign a water depth or maybe even assign a temperature range. And as you get more specific, your confidence is going to decrease. So at what point do they become unacceptable? At what point are these analog interpretations unacceptable? And so, for example, I can't show you that Pliocene or that planktic foraminiferous species have identical preferred surface temperatures as they do today. But I can tell you that the same species is abundant in the same temperature range today as it was in the Pliocene. So I guess my question is that good enough? And the final slide. So in conclusion, so we know that the Planktiform species abundance is mostly tied to sea surface temperature. And we've shown that in the species that we have investigated, which is only a few, sea surface temperature responses do appear similar between the Pliocene and the modern. But we've learned from some of the previous discussions today that some species maintain this same temperature response, but some species don't. And of course, this depends on the stability of the alkaline paleothermometer over time, which is a question. But mostly it depends on the definition of similar. How similar do we need to be? So for our purposes, Planktiform temperature preferences are stable enough to support the continued use of our assemblage-based temperature proxies. And we found also that they're pretty in close agreement with other proxies that are completely independent. And that's it. Thank you.
That's great. Thank you, Marcy. I'm glad we got that working.
Yeah.
Let's have a look for questions. I think I saw some questions popping in. Down to get on the chat. Where have I gone? There we go. We have a question coming through. Do we think, or do you think the species are genetically adapted to different sea surface temperatures through time? Is there a genetic component in here?
Well, that's sort of beyond what we do. I mean, we don't look at the genetics at all. There certainly could be. I mean, there's been a lot of genetic work happening that shows differences, but we're paleontologists. We're looking at what the tests look like. There could be. I don't have any information on that, but certainly there could be.
And how confident are you that your Pliocene forams really are conspecific with the modern species? So obviously, I guess it's a morphological concept again, isn't it? But
yeah, it is. Well, I mean, there's been a lot of work done on the species that we were talking about, the one species that's still extant from the Pliocene, Incomta, and it does seem to go back 1 million years. The others, I'm not sure there has been work on that. But like we were talking about, some of these aren't the same species. We have made a bunch of groupings. And even the ones that have evolved, they've evolved into new species. But what we're really looking at is whether they occupy the same temperature salinity space in the Pliocene as they do today. And if they do, then we're making some assumptions there.
Yeah, so just following up from that, you mentioned about you're looking forward to just getting that northern site that you've now just received the data for, obviously to extend, some colder water data there. Are there any other sites or locations where you really wish you had data, or the next sort of data point that you really want to include for the modeling you're doing?
Yeah, well, you've seen our, I mean, I showed the map of where our sites are, and they're pretty well globally distributed. We have what I showed was the North Atlantic, because that, of course, is where we have the highest concentration of them. And also we have really good age models. So, so we can look at those samples and compare them to each other. We could look for more. We've gotten more in the North Atlantic, and we have more samples in the Bay of Bengal now too. And the Bay of Bengal is interesting because it does have a salinity component there, which is changing things. So yeah, that would be an example of some place where we would go to look at something other than temperature that's changing. And then those two sites that I showed you in the North Atlantic, that something definitely other than temperature is happening. We'll look harder at those to see what's going on.
Yeah. Well, we've got another question from Sierra Peterson. It seems like a strength of this method that you look at many species of forams. Do you ever look at whether the groups hold are consistent, I guess, or whether individuals cease to follow their groupings? So sort of within group deviations, I guess.
We haven't yet. So there are a lot of questions now. So again, we have this amazing database of sample over 1 million. We have over 1 million planktic forams that we've looked at from this very restricted period. And the PRISM project, which is had been going on for 30 years, was gathering this data for one specific purpose. So, but what we've been doing the past couple of years is going back and looking to see what else this amazing data set can offer us. So we haven't looked at that yet, but that is one thing that we can go back and start looking at. And, I only showed you 4 different species associations, and that's because that's all we've looked at so far. We've got many more species to look at.
Great. And sort of following on a bit, I guess, Carl Flesser likes the question good enough. So of course, is everything good enough for what we want to do with our data? So he asks, what is the goal here? Is it to match the model temperatures? And is there a concern over niche stability? Is this concern over niche stability another manifestation of our occupation concern over the inadequacy of the fossil record? Deep question to finish with there, I think.
So we have a really fun relationship with modelers, and we never try to match model temperatures. That's not how we look at that. But the modelers do want the least bit of error possible, right? That's what they want from us. Which is not always, because we work with fossils, right? That's not always easy to get. And and you can figure out what your analytical error is, but it's harder to figure out what your error is when you're looking at fossil assemblages. So we're always going to have more error than the modelers want. And gosh, I forget the second part of that question, but could you read that again?
Yeah, sorry. Is this concern, our general concern over niche stability, is it another manifestation of our occupation concern over the inadequacy of the fossil record?
Well, we are lucky enough to have lots and lots and lots of planktic forams. I mean, if you're gonna, in terms of fossil records, it's a good one to work with, because there are a lot of them and they're everywhere. Yeah, I don't I think that was just a comment. I'm not quite sure how to answer that.
That's fine. I think that's a general one, isn't it, for a discussion carrying on, or for another time. Thanks for that one, Carl. Thanks, Marcy. That was great. I think I might be handing back now to Andy. The end now, I think. Is that right? Andy's going to turn his microphone on and tell me if that's the case or not.
And thank you again, Becky, for fixing that for me.
No problem.
Okay. Am I coming through okay now? Okay, well, thank you to all speakers. Thank you to Richard and Liz for so expertly chairing the two sessions. Thank you to Becky for dealing with the logistics and also finding a solution to that last minute hitch, the sort of thing that one fears all the time in these kind of events, and for enabling Marcy to get through so well. So thank you all, and thank you all, of course, those who have attended as the audience. I think the meeting has succeeded to the extent that those that have attended are diverse in their interests. They come from a wide diversity of fields, and so it succeeded in bringing those people together and talking to each other and sharing their knowledge and interests in a way that I suspect hasn't happened. You know, there are perhaps people who have learned about fields that are not particularly familiar with. I certainly have, but the meeting will really only be a success if that contact and that sharing of information and that mutual interest is continued. And so what we're looking to do is to have a meeting in a couple of years' time, a full conference, maybe not necessarily an in-person conference, maybe another virtual event, that remains to be decided, but a larger scale event at which we hope new data, perhaps stemming from collaborations and ideas stimulated by this meeting, which the new results will be presented and new thinking will be presented as well. And provisionally, the JOLSOC has said that they will badge that meeting, possibly co-badge that meeting with the Natural History Museum. But we'd like to know, obviously, whether there's going to be sufficient interest for that event to take place. So what I've asked Becky to do in the next day or two is to send out a circular to all those who registered for this event, asking them to give an expression of interest, basically, in whether they would like to attend such an event in a couple of years' time, 2024, and possibly present. Absolutely non-binding indication, but that will at least give us some idea of whether this is something that we want to take forward. I think if you perhaps were of the view that niches were immutable, you may have had your preconceptions altered a little bit by this meeting. Certainly there are some indications of niche change, and what you've heard is something about the methods by which change might be identified, by looking at mutual occurrence of species, by looking at independent evidence in the form of geochemical data and so on and so forth. And as you can see, that's going on, and there's obviously there's plenty of potential for more of that to go on, and hence for the whole question of niche stability to be addressed on the wide scale that it needs to be. So thank you again for your attendance. If you missed any of the meeting, or perhaps just didn't keep up with what was being presented, there will be a YouTube recording available via the JOLSOC's channel in a day or two, I understand. So you can check back on that to pick up details that you may have missed. So on behalf of the conveners, Becky, and everybody else, thank you very much for attending. And I hope very much that we can put on some sort of a similar but larger event in a couple of years' time. Thank you very much.
Thank you all.
Thank you, Andy. Thank you. Bye.
Thanks
Thank
all.
you, everybody.
Cheers.
Very interesting stuff. I've
Bye,
learned a few
everyone.
things today. Thank you for coping with that, Marcy, as well. You did very, very well.
Oh, thanks.
And
The
thanks for everyone for kind of, the time zone differences and getting it all right. So really appreciate that. Thank you.
Yeah, it went very smoothly.
All right, I'll close things down now, but it will be live on YouTube probably tomorrow.
Okay. And we'll perhaps be in touch, Becky, to just discuss what sort of form of words we'll send around to people to get some idea of what interest there would be. Obviously, it's only a small sample, but it's a representative sample, perhaps. And, we can maybe